Hello and welcome to Nighttime Science. Imagine lying very still in a darkened room, the curtains drawn, the air settling into a gentle, steady quiet. Somewhere outside that room, perhaps just beyond the glass of a window, a leaf is slowly rotating to catch the last of the evening light. A branch is easing downward by a few millimeters as internal water pressure shifts. Tiny pores on the surface of a leaf are narrowing, preparing for the long stretch of night. These motions are so gradual that they slip past human awareness, yet they are continuous, persistent, and deeply orderly, governed by the unhurried science of plant life. Plants and trees move on timescales that are almost the opposite of a racing human mind. Where thoughts jump and flicker, a plant leans toward the light over hours, or adjusts its internal chemistry in response to a passing cloud. Their world is filled with slow currents and microscopic events. Ions crossing membranes, water columns rising through slender tubes, packets of sugar traveling from leaf to root. When seen through the lens of biology, this quiet world becomes a collection of gentle processes that can be followed step by step, like watching a slow tide move in and out along a shoreline. In the stillness of nighttime, the science of plants and trees unfolds in particularly soothing ways. During the day, a leaf is busy catching photons, shuttling electrons, building energy-rich molecules. At night, many of those processes shift into a calmer mode as the plant focuses on respiration, repair, and growth. Some trees even change their posture. Certain birch trees have been observed lowering their branches during the night and raising them again by morning, a phenomenon that suggests a kind of daily resting rhythm tied to internal water status and subtle changes in cell pressure. The story of plants is a story of patience, of steady cycles that repeat day after day and year after year. Each tree in a forest has a biography written into its rings, each ring representing a year of growth, with widths that reflect seasons of plenty or hardship. Even the air we breathe carries the long legacy of microscopic plant-like organisms that began releasing oxygen billions of years ago, steadily changing the composition of Earth's atmosphere. The modern green leaf continues this work, turning light, carbon dioxide, and water into sugars, and releasing oxygen as a byproduct. There is also a hidden social dimension to plant life that lends itself to a quietly fascinating exploration. Beneath many forests lies a network of fungal threads, thin filaments that connect the roots of multiple plants into shared pathways for nutrients and signals. Through these connections, trees can transfer carbon, nitrogen, and other compounds, and can even send warning signals when pests or diseases threaten. This underground exchange is not dramatic in the way human communication can be. It is gradual, chemical, and diffuse. Yet it reveals that a forest is more than a collection of isolated trunks. It is a connected system, quietly coordinating in ways that invite careful, unhurried attention. The inner workings of a single leaf also provide a soft, intricate subject to rest the mind on. Inside plant cells, chloroplasts house the molecular machinery of photosynthesis, where pigments absorb light and use it to drive a series of reactions that store energy in chemical bonds. These reactions proceed through well-characterized steps. Water is split, oxygen is released, and molecules like ATP and NADPH are produced, which then power the building of sugars from carbon dioxide. Each step is small and precise, like a long chain of tiny switches turning on in sequence, never rushed, always following the same calm pattern. Plant science also includes many small, gentle forms of motion that become evident when viewed over longer periods. The slow bending of a stem toward a window is an example of phototropism, where growth on one side of the stem is encouraged by a hormone called auxin, causing the plant to lean toward the light source. Roots grow downward through soil in response to gravity, guided by specialized cells and hormone gradients in a process known as gravitropism. These growth patterns are careful adjustments rather than sudden movements, and thinking about them can give the mind something steady and predictable to follow. Even more subtle are the changes plants make to manage gases and water. On the surfaces of leaves, stomata, tiny adjustable pores bordered by pairs of guard cells, open and close to balance carbon dioxide intake with water loss. During bright daylight, many plants open their stomata wider to allow more carbon dioxide in for photosynthesis, while at night these pores often close more tightly, conserving water when light is not available for sugar production. This quiet regulation happens continuously, day after day, as each leaf responds to humidity, light levels, and internal signals. In forests where tall trees rise high into the air, the internal flow of water and nutrients follows predictable laws of physics. Water absorbed by roots travels upward through xylem, pulled by the combination of evaporation at the leaves and the cohesion and adhesion of water molecules. Sugars produced in leaves are distributed through phloem, moving from areas of higher concentration, such as the leaves, to areas of lower concentration, such as growing roots or developing fruits. These flows are steady and quiet, like unseen rivers moving inside a trunk. Plants occupy nearly every environment on Earth, from dense rainforests to dry deserts. Some have formed partnerships with bacteria that can convert atmospheric nitrogen into forms that plants can use, a process known as biological nitrogen fixation. This symbiosis, especially common in legumes like peas and beans, allows these plants to thrive in soils that might otherwise lack sufficient nitrogen, slowly enriching the soil over time. Elsewhere, carnivorous plants supplement their nutrient supply by capturing insects, using specialized leaves that can close or trap prey, powered by carefully orchestrated changes in cell turgor and structure. Across centuries, some trees persist as living witnesses to environmental change. Certain bristlecone pines have been dated to several thousand years in age, making them among the oldest known individual organisms. Their growth rings record periods of drought, cooler years, and other climatic variations, allowing scientists to reconstruct aspects of past climates and events such as extended dry spells or volcanic eruptions. High above, some coast redwoods and giant sequoias reach heights well over 90 meters, relying on structural strength and specialized water transport to sustain their towering forms. This script will wander slowly through these and many other details of plant and tree biology, pausing over each one long enough to make it familiar and gently vivid. The focus will remain on calm, factual observations: how trees communicate underground, how leaves manage light and air, how roots explore soil, how seeds wait for the right moment to emerge. The goal is to let the mind rest on these quiet processes, to feel how measured and continuous they are, and to let that sense of continuity support relaxation. As the exploration continues, the pacing will remain unhurried. The narrative will linger on the way water moves, how gases diffuse, how a forest canopy shapes the air beneath it. It will move from the secret language of trees beneath the soil, to the sleeping posture of branches in the dark, to the patient life of the tallest and oldest trees. With each step, the aim is to provide a gentle stream of grounded scientific detail that can slowly fade into the background as sleep approaches, while still remaining accurate and complete for anyone who chooses to listen all the way through. At the end of this long journey through plant and tree science, there will be a soft closing, with a quiet reminder that if this slow exploration has helped you feel calmer, you are welcome to return, to listen again, and to support the channel with a like or a subscription so that more soothing scientific journeys can be created for you. In a forest, what appears to be a collection of separate trees standing in soil is in many cases something far more interconnected. Beneath the visible surface, a vast and intricate network of fungal filaments weaves through the earth, linking root to root, tree to tree, creating pathways for the exchange of nutrients, water, and chemical signals. This underground system, often referred to as a mycorrhizal network, operates continuously and quietly without any need for conscious direction, forming what some researchers have informally called the Wood Wide Web. Mycorrhizae are symbiotic associations between fungi and plant roots. The word itself comes from Greek roots meaning fungus and root. In these partnerships, the fungus colonizes the root system of a host plant, either by penetrating the root cells or by forming a sheath around them. In exchange for sugars that the plant produces through photosynthesis, the fungus provides the plant with improved access to water and nutrients, particularly phosphorus and nitrogen, which can be difficult for roots to obtain on their own. The fungal threads, called hyphae, are much finer than plant roots and can explore soil more thoroughly, reaching into tiny crevices and extending the plant's effective absorption area by a significant degree. These fungal networks do not connect just one plant to one fungus. A single fungal individual can link with multiple plants, and a single plant can be colonized by multiple species of fungi. The result is a web of connections that can span across entire forest floors, creating a shared underground infrastructure. Through this network, carbon compounds produced by one tree can move to another tree, sometimes even a tree of a different species. Research has shown that larger, older trees with access to more sunlight can transfer carbon to smaller, younger trees growing in shoddier conditions, effectively subsidizing their growth. The mechanisms by which this transfer occurs are rooted in basic principles of diffusion and active transport. Sugars produced in the leaves of a donor tree travel down through the phloem to the roots, where some of those sugars are exchanged with the mycorrhizal fungi. The fungi, in turn, can transport these carbon compounds through their high-fall networks to other locations, including the roots of neighboring plants. At the receiving end, the sugars can be absorbed by the roots of another plant, supplementing that plant's own photosynthetic production. This process is not instantaneous, but occurs steadily over days and weeks, with the flow of resources adjusting based on the relative needs and availabilities within the network. Beyond carbon, mycorrhizal networks also facilitate the transfer of nitrogen and phosphorus. Nitrogen, which is essential for building proteins and nucleic acids, can be captured from decaying organic matter by fungi and delivered to plant roots. Phosphorus, often locked in mineral forms that roots cannot easily access, can be mobilized by fungal enzymes and absorbed by the hyphae, then passed along to the plant. In this way, the fungal network acts as an extended foraging system, greatly increasing the efficiency with which plants can gather essential elements from the soil. Interestingly, these networks also appear to carry information. When a plant is attacked by insects or infected by a pathogen, it can produce chemical signals both above ground and below. Some of these signals are volatile organic compounds released into the air, which can warn neighboring plants of danger and prompt them to activate their own defenses. Below ground, chemical signals can also travel through mycorrhizal networks. Studies have demonstrated that plants connected by a shared fungal network can exhibit defensive responses even before they themselves are attacked, suggesting that warning signals have traveled through the soil from an affected neighbor. The nature of these signals is chemical and diffuse. A plant under stress might alter the composition of the compounds it releases into the soil or exchanges with its fungal partners. These changes can include increases in certain hormones or secondary metabolites that indicate the presence of herbivores or pathogens. The fungi, acting as conduits, transport these compounds along with nutrients, and when they reach the roots of another plant, that plant's biochemical detection systems can recognize the change and respond by upregulating defensive genes, producing toxins or deterrents, or strengthening cell walls. This form of communication does not involve intention or awareness in any conscious sense. It is an emergent property of interconnected biochemical systems. The fungi are not deliberately carrying messages, they are simply moving compounds as part of their metabolic activity. The plants are not consciously warning each other, they are responding to chemical cues in their environment according to genetic programming shaped by evolution. Yet the outcome is a kind of distributed information network, where the state of one part of the forest can influence the state of another part, creating a system that is more resilient and responsive than any single tree could be on its own. The existence of mycorrhizal networks has implications for how forests recover from disturbances. When a tree is damaged or dies, the nutrients it held in its tissues can be redistributed through the fungal network to surviving trees, rather than being lost entirely. Seedlings establishing in a forest can tap into an existing network, gaining access to resources and information that would otherwise take much longer to acquire. In this sense, the network provides a form of continuity and memory, carrying resources and potentially even defensive information across generations of trees. Not all plants participate equally in these networks. Some species are more heavily colonized by mycorrhizal fungi than others. In general, trees and shrubs in temperate and boreal forests form extensive mycorrhizal associations, particularly with ectomycorrhizal fungi that form sheaths around root tips. In contrast, many agricultural crops and some pioneer species have less developed mycorrhizal relationships or rely on different types of fungi. This variation means that the structure and density of underground networks can differ greatly depending on the plant community and soil conditions. The study of these networks is still revealing new details. Recent research has begun to map the architecture of mycorrhizal networks in specific forests, identifying hub trees that are especially well connected and that may play disproportionate roles in nutrient distribution. Other studies are exploring how environmental changes such as shifts in temperature or soil moisture affect the functioning of these networks. There is also interest in understanding how human activities such as logging or soil disturbance disrupt fungal networks and what that means for forest health and regeneration. For anyone lying quietly and listening to this description, the image of an underground web of fine threads pulsing slowly with the movement of nutrients and signals can serve as a calming focal point. The scale is vast, but the processes are gradual. Carbon atoms produced in a sunlit leaf travel down through living tissue, cross into a fungal cell, move along a thread-like hypha for meters or even tens of meters, and then cross into the root of another tree. Each step follows predictable chemical gradients and transport mechanisms. There is no urgency, only a steady flow governed by concentration differences and active pumps in cell membranes. In thinking about this hidden dimension of forest life, it becomes clear that the soil is not merely an inert substrate. It is an active, living medium filled with microscopic partnerships and exchanges. The fungi themselves are ancient organisms with lineages stretching back hundreds of millions of years, long before flowering plants appeared. The mycorrhizal symbiosis is thought to have been critical in allowing early plants to colonize land by helping them extract nutrients from rocky, nutrient-poor soils. That partnership has persisted and diversified, becoming a foundational feature of most terrestrial ecosystems. The quiet persistence of these fungal networks mirrors the quiet persistence of the trees themselves. Both operate on timescales that are slow relative to human experience. A mycorrhizal network can take years to fully develop as fungi colonize roots and extend through the soil. A tree can live for decades, centuries, or even millennia, continuously participating in nutrient exchanges with its fungal partners. Throughout that long life, the tree both gives and receives, contributing carbon when it has surplus and drawing on shared resources when it is stressed or young. This reciprocal relationship extends to the fungi as well. While the plant provides sugars, the fungus provides not only nutrients but also a degree of protection. Mycorrhizal fungi can help buffer plants against drought by improving water uptake, against toxic metals by sequestering them in fungal tissues, and against certain soil pathogens by occupying space on the root surface and producing defensive compounds. The partnership is mutually beneficial and stable over long periods, maintained by evolved mechanisms that balance costs and benefits for both organisms. As the mind follows these underground pathways, there is a gentle rhythm to the concepts. Hyphae extending, nutrients flowing, signals diffusing, trees responding. Each element can be pictured slowly without rush. The image of a forest floor covered in leaf litter with an invisible lattice of fungal threads beneath creates a sense of hidden depth and complexity that is nevertheless orderly and comprehensible. The science here is not speculative. It is based on careful experiments involving isotopic tracers, careful excavation of root systems, and measurements of nutrient flows. The conclusions are supported by data, yet they invite a kind of quiet wonder at the hidden cooperation that sustains the visible world of trunks and leaves. The idea that trees might sleep seems at first like a poetic metaphor rather than a scientific observation. Yet research has revealed that many trees do exhibit daily rhythms that resemble sleep in certain respects. These rhythms involve measurable changes in branch position, leaf orientation, and internal physiology that occur on a roughly 24-hour cycle, synchronized with the alternation of day and night. While trees do not have nervous systems or brains and therefore do not sleep in the way animals do, they do show clear evidence of circadian regulation, a biological timekeeping mechanism that coordinates many aspects of their metabolism and growth. One of the most striking demonstrations of tree sleep came from a study conducted by researchers in Finland and Austria, who used laser scanning to measure the position of birch tree branches over the course of a night. The measurements were taken in controlled conditions with no wind, to ensure that any observed movement was due to the trees themselves rather than external forces. The results showed that the branches and leaves of the birch trees drooped by as much as 10 centimeters during the night, with the lowest point occurring a few hours before dawn. As morning approached and light returned, the branches gradually lifted back to their daytime position. This cyclical droop and recovery occurred consistently, suggesting an internal process tied to the tree's circadian rhythm. The mechanism behind this nighttime drooping is thought to involve changes in turgor pressure within the cells of the tree. turgor pressure is the force exerted by water inside plant cells pushing against the cell walls. When cells are fully hydrated, they are firm and help support the structure of leaves and branches. When water pressure decreases slightly, cells become less rigid and the tissues they form may sag or droop. During the day, active photosynthesis and transpiration maintain high water flow through the tree, keeping cells turgid. At night, when photosynthesis stops and transpiration slows, the water dynamics shift, and some tissues may lose a small amount of turgor, leading to the observed droop. This change is not simply a passive response to the absence of light. Trees possess internal circadian clocks, molecular systems that oscillate with a period of approximately 24 hours even in the absence of external time cues. These clocks regulate the expression of thousands of genes, controlling processes such as growth, hormone production, and stress responses. In many plants, the circadian clock influences the opening and closing of stomata, the timing of flower opening, and the sensitivity to light and temperature. The nighttime drooping of branches appears to be another output of this clock, a way for the tree to adjust its posture in coordination with the daily cycle. The biological advantage of such a rhythm is not entirely clear, but several hypotheses have been proposed. One possibility is that reducing leaf and branch exposure at night minimizes water loss during a period when photosynthesis is not occurring and when humidity is often higher, reducing the need for transpiration-driven water uptake. Another possibility is that the droop reflects a reallocation of resources or a shift in metabolic priorities, with the tree directing energy toward growth, repair, or storage rather than maintaining maximal structural rigidity. A third idea is that the circadian rhythm helps the tree anticipate the coming dawn, preparing its tissues to resume photosynthesis efficiently as soon as light is available. Regardless of the precise evolutionary advantage, the phenomenon illustrates that trees are dynamic organisms, constantly adjusting their physiology in response to internal and external signals. The daily cycle of droop and recovery is just one example of the many rhythms that govern plant life. Other examples include the rhythmic opening and closing of flowers, the timing of leaf unfolding in spring, and the seasonal cycles of growth and dormancy. All of these are coordinated by overlapping systems of hormones, light receptors, and clock genes. At the molecular level, the circadian clock in plants involves feedback loops of gene expression. Certain clock genes are turned on by light or other signals, and the proteins they produce accumulate over time. These proteins then inhibit their own production, causing their levels to decline, which eventually allows the genes to be turned on again, restarting the cycle. This oscillation takes roughly 24 hours to complete and is remarkably stable, persisting even in constant darkness or constant light, though it can be reset by environmental cues such as dawn or dusk. The clock influences a wide range of downstream processes. For example, genes involved in photosynthesis are often upregulated in anticipation of dawn, so that the photosynthetic machinery is ready to capture light as soon as the sun rises. Similarly, genes involved in starch breakdown may be upregulated at night, ensuring a steady supply of sugars to support respiration and growth when photosynthesis is not occurring. This anticipatory regulation allows the plant to be more efficient than it would be if it simply reacted to changes in light after they occurred. The nighttime metabolism of trees differs in important ways from their daytime metabolism. During the day, photosynthesis in the leaves produces far more sugars than the plant needs for immediate energy, and the excess is stored as starch in the chloroplasts. At night, photosynthesis ceases because there is no light to drive the light-dependent reactions. The plant must then rely on cellular respiration to generate the ATP needed for maintenance, growth, and other processes. Cellular respiration occurs in the mitochondria and involves breaking down stored carbohydrates, using oxygen, and releasing carbon dioxide, water, and energy. Interestingly, plant respiration at night is not solely determined by temperature as was once widely believed. Recent research has shown that nocturnal respiration rates are under strong biological control and can vary independently of temperature, influenced by factors such as the plant's circadian clock, its developmental stage, and its recent history of carbon gain. This suggests that plants actively regulate how much of their stored resources they use at night, balancing the need for immediate energy against the need to conserve reserves for future use. Some plants have evolved specialized forms of metabolism that alter the typical day-night pattern. For example, plants that use crassulacean acid metabolism, or CAM, open their stomata at night instead of during the day, taking in carbon dioxide in the dark and storing it as organic acids. During the day, the stomata close to conserve water, and the stored carbon dioxide is released internally and used for photosynthesis. CAM plants, which include many succulents and cacti, show elevated nocturnal respiration rates related to the energy costs of this carbon storage process. This adaptation allows them to photosynthesize while minimizing water loss in arid environments, demonstrating yet another way that plants adjust their rhythms to match their ecological circumstances. For most trees in temperate and tropical forests, however, the night is a time of reduced activity compared to the day. Transpiration slows as stomata close or narrow, reducing water loss. Photosynthesis halts and the focus shifts to respiration and growth. Many trees grow primarily at night or in the early morning when temperatures are cooler and water stress is lower. Cell division and elongation, which require energy and building materials, draw on the sugars and starches accumulated during the previous day. This nocturnal growth is another aspect of the tree's daily rhythm, invisible to casual observation but measurable with sensitive instruments. The concept of trees sleeping can also extend to longer time scales. In temperate regions, many trees enter a state of dormancy during winter, shedding their leaves and ceasing active growth. This dormancy is not the same as sleep, but it shares some features including a reduction in metabolic activity and responsiveness to external stimuli. Dormancy is triggered by decreasing day length and cooler temperatures, which signal to the tree that winter is approaching. The tree then undergoes a series of physiological changes, including the breakdown of chlorophyll in the leaves, the sealing off of leaf attachment points, and the hardening of tissues to withstand freezing. During winter, the tree's metabolism slows dramatically, and it relies on stored reserves to survive until spring. Even during this dormant period, some processes continue. Roots may still absorb small amounts of water and nutrients if the soil is not frozen. Buds remain alive, protected by specialized scales and antifreeze compounds that prevent ice crystal formation inside cells. The circadian clock continues to run, though its outputs may be suppressed or redirected. When spring arrives and day length increases, the clock helps coordinate the resumption of growth, triggering bud break and the unfolding of new leaves in a carefully timed sequence. The daily and seasonal rhythms of trees are thus deeply intertwined. The circadian clock provides a daily framework that is modulated by seasonal signals, allowing the tree to adjust its behavior over multiple timescales. This layered regulation is essential for survival in environments where conditions change predictably over the course of a day and a year. By anticipating these changes and preparing in advance, trees can maximize their growth and resource acquisition while minimizing their exposure to stress. For a listener resting in the quiet, the image of a tree slowly lowering its branches as night deepens, then slowly raising them again as dawn approaches, offers a gentle, repetitive motion to contemplate. The process is automatic, driven by internal timers and subtle shifts in water pressure, yet it mirrors the rhythms of animal sleep and waking. There is something calming in the thought that even a tree rooted in one place and without a nervous system participates in the daily cycle of activity and rest, adjusting its posture and metabolism in harmony with the turning of the earth. Photosynthesis is the process by which plants capture light energy and transform it into chemical energy stored in the bonds of sugar molecules. It is one of the most important biochemical processes on Earth, responsible for producing the oxygen in the atmosphere and forming the base of nearly all food chains. Yet for all its global significance, photosynthesis unfolds quietly within microscopic structures inside plant cells, following a sequence of steps that can be traced molecule by molecule, photon by photon. The primary sites of photosynthesis are chloroplasts, specialized organelles found in the cells of leaves and other green tissues. Each chloroplast is enclosed by a double membrane and contains an internal system of flattened sacs called thylakoids, which are stacked in some regions to form structures known as grana. Embedded in the thylakoid membranes are the pigments and proteins that capture light and carry out the initial steps of photosynthesis. The fluid surrounding the thylakoids, called the stroma, contains enzymes that catalyze the later steps of the process, where carbon dioxide is incorporated into organic molecules. Photosynthesis can be divided into two main stages: the light-dependent reactions and the light-independent reactions, also known as the Calvin cycle. The light-dependent reactions occur in the thylakoid membranes and require light energy. The Calvin cycle occurs in the stroma and does not directly require light, though it depends on the products of the light-dependent reactions. Together, these stages convert light energy, water, and carbon dioxide into glucose and oxygen. The light-dependent reactions begin when photons of light strike pigment molecules in the thylakoid membrane. The most important of these pigments is chlorophyll, which absorbs light most efficiently in the blue and red portions of the spectrum and reflects green light, giving leaves their characteristic color. When a chlorophyll molecule absorbs a photon, one of its electrons is excited to a higher energy level. This high-energy electron is then transferred to a nearby molecule, beginning a chain of electron transfers that ultimately stores energy in chemical form. There are two main protein complexes involved in the light-dependent reactions: photosystem II and photosystem I, often abbreviated as PS2 and PS1. Despite their names, photosystem II acts first in the sequence. When light excites electrons in the chlorophyll of photosystem II, those electrons are passed to an electron transport chain. To replace the lost electrons, photosystem II splits water molecules in a reaction that releases oxygen gas as a byproduct. This splitting of water is the source of almost all the oxygen in Earth's atmosphere, a process that has been occurring for billions of years, ever since photosynthetic organisms first evolved. The electrons from photosystem II move through a series of carrier molecules in the thylakoid membrane, losing energy at each step. Some of this energy is used to pump hydrogen ions, also called protons, from the stroma into the interior of the thylakoid, creating a concentration gradient. This gradient represents stored potential energy, much like water held behind a dam. Eventually, the electrons reach photosystem 1, where they are re-energized by another absorption of light. From photosystem 1, the electrons are transferred to a molecule called NADP, forming NADPH, a carrier of high-energy electrons. Meanwhile, the hydrogen ion gradient across the thylakoid membrane is used to generate ATP, the universal energy currency of cells. An enzyme called ATP synthase spans the thylakoid membrane and acts as a molecular turbine. As hydrogen ions flow back through this enzyme down their concentration gradient, the energy of their movement is harnessed to attach a phosphate group to ADP, forming ATP. This process, known as chemiosmosis, is remarkably efficient and is also used in mitochondria during cellular respiration. At the end of the light-dependent reactions, the chloroplast has produced two key molecules, ATP and NADPH. Both are rich in energy and will be used in the next stage of photosynthesis to build sugars from carbon dioxide. Oxygen, the other product of the light-dependent reactions, diffuses out of the chloroplast and eventually out of the leaf, entering the atmosphere or being used by the plant's own mitochondria for respiration. The Calvin cycle, which takes place in the stroma of the chloroplast, is a series of enzyme-catalyzed reactions that fix carbon dioxide into organic molecules. The term fix here means to convert an inorganic form of carbon dioxide gas into an organic form that can be used to build sugars, amino acids, and other compounds. The cycle begins when carbon dioxide from the air enters the leaf through stomata and diffuses into the stroma. There, an enzyme called rubisco, short for ribulose 1,5-bisphosphate carboxylase oxygenase, catalyzes the attachment of carbon dioxide to a 5-carbon sugar called ribulose bisphosphate. This reaction produces an unstable 6-carbon compound that immediately splits into 2 molecules of a 3-carbon compound called 3-phosphoglycerate. These 3-carbon molecules are then converted through a series of steps involving ATP and NADPH into another 3-carbon molecule called glyceraldehyde 3-phosphate, or G3P. Some of the G3P molecules are used to regenerate ribulose bisphosphate, allowing the cycle to continue. The remaining G3P molecules are the output of the Calvin cycle and can be used to build glucose and other carbohydrates. The Calvin cycle must turn 6 times, fixing 6 molecules of carbon dioxide to produce 1 molecule of glucose, which contains 6 carbon atoms. This requires the input of 18 molecules of ATP and 12 molecules of NADPH, all of which must be supplied by the light-dependent reactions. The process is not instantaneous. Each turn of the cycle involves multiple enzyme-catalyzed steps, and the entire sequence proceeds at a steady, measured pace determined by enzyme kinetics and the availability of substrates. Rubisco, the enzyme that catalyzes the first step of the Calvin cycle, is thought to be the most abundant protein on Earth, reflecting the importance of carbon fixation to life. However, rubisco is not particularly efficient. It sometimes mistakenly binds oxygen instead of carbon dioxide, leading to a wasteful process called photorespiration. Despite this inefficiency, rubisco has been retained throughout the evolution of plants, and various adaptations have evolved to minimize the impact of photorespiration, including specialized leaf anatomy in some plants and the CAM metabolism mentioned earlier. The sugars produced by the Calvin cycle serve multiple functions in the plant. Some are immediately used for energy through cellular respiration. Some are converted into starch and stored in the chloroplasts or other parts of the plant, such as roots or tubers, for later use. Some are used as building blocks to synthesize cellulose for cell walls, proteins for enzymes and structures, lipids for membranes, and nucleic acids for DNA and RNA. In this way, the simple act of capturing light and fixing carbon provides the raw materials and energy for all of the plant's growth and maintenance. The efficiency of photosynthesis varies depending on conditions such as light intensity, temperature, carbon dioxide concentration, and water availability. Under ideal conditions, a plant can convert a small percentage of the light energy it receives into chemical energy stored in sugars. This may seem modest, but over the course of a growing season, the cumulative effect is substantial, allowing plants to build large structures and accumulate significant biomass. Plants have evolved various adaptations to optimize photosynthesis under different environmental conditions. In low light, some plants produce more chlorophyll or adjust the size and number of their chloroplasts to capture as much light as possible. In high light, excess energy can damage the photosynthetic machinery, so plants have protective mechanisms, such as dissipating excess energy as heat, or producing antioxidant molecules to neutralize harmful reactive oxygen species. The color of light also matters. Chlorophyll absorbs red and blue light most effectively, while green light is largely reflected. This is why leaves appear green to the human eye. However, plants also contain accessory pigments, such as carotenoids, which absorb light in different parts of the spectrum and transfer that energy to chlorophyll, broadening the range of light that can be used for photosynthesis. Carotenoids are responsible for the yellow and orange colors seen in some leaves, particularly in autumn, when chlorophyll breaks down and reveals the underlying pigments. The process of photosynthesis is thus a finely tuned system, with multiple feedback loops and regulatory mechanisms ensuring that the plant makes the best use of available light while avoiding damage. The thylakoid membranes are packed with protein complexes and pigments arranged in precise spatial configurations to facilitate efficient energy transfer. The enzymes of the Calvin cycle are regulated by light, pH, and the availability of substrates, ensuring that carbon fixation occurs when energy and reducing power are available. For a mind-seeking calm, the step-by-step nature of photosynthesis offers a soothing mental pathway. Light strikes a leaf, a photon is absorbed, an electron is excited. The electron moves through a chain of carriers. Water is split, oxygen is released, hydrogen ions flow through ATP synthase. ATP is made, carbon dioxide is captured, a sugar is built. Each of these events follows laws of chemistry and physics, proceeding in an orderly sequence that has been repeated countless times in countless leaves over hundreds of millions of years. The constancy and reliability of this process, the quiet transformation of light into life, can serve as a gentle anchor for drifting thoughts. While photosynthesis captures the attention as the signature process of plant life, plants also engage in cellular respiration, the same fundamental metabolic pathway used by animals and most other organisms to extract energy from organic molecules. Respiration occurs continuously in plants, both day and night, in all living cells, providing the ATP needed to power growth, transport, synthesis, and maintenance. At night, when photosynthesis ceases, respiration becomes the plant's sole source of metabolic energy, drawing on stored carbohydrates to sustain the organism until light returns. Cellular respiration in plants takes place primarily in the mitochondria, organelles that are structurally and functionally similar to those found in animal cells. The overall equation for respiration is essentially the reverse of photosynthesis. Glucose and oxygen are consumed, and carbon dioxide, water, and energy are released. However, the detailed biochemistry involves a complex series of reactions that extract energy in small, controlled steps, capturing it in the form of ATP rather than releasing it all at once as heat. Respiration can be divided into three main stages: glycolysis, the citric acid cycle, also known as the Krebs cycle, and oxidative phosphorylation, which includes the electron transport chain, and chemiosmosis. Glycolysis occurs in the cytoplasm and breaks down glucose into two molecules of pyruvate, producing a small amount of ATP and NADH in the process. The pyruvate then enters the mitochondria, where it is further broken down in the citric acid cycle, releasing carbon dioxide and generating more NADH and FADH2, both of which are carriers of high-energy electrons. Finally, the electron transport chain in the inner mitochondrial membrane uses these electrons to pump protons and create a gradient, which drives the synthesis of ATP through ATP synthase, much as occurs in the thylakoid membranes during photosynthesis. The rate of plant respiration is influenced by many factors, including temperature, the availability of oxygen and substrates, and the plant's developmental stage. For many years, it was assumed that nighttime respiration rates in plants were determined primarily by temperature, with warmer nights leading to higher respiration and greater carbon loss. However, recent research has revealed that nocturnal plant respiration is under strong biological control and does not always follow simple temperature relationships. Instead, respiration rates at night are modulated by the plant's circadian clock, its recent history of photosynthesis, and internal signals related to carbon status and growth demands. This biological regulation means that plants can adjust how much of their stored carbon they consume at night, balancing the need for energy against the need to conserve resources for future growth or stress responses. In some cases, plants may reduce nighttime respiration rates even when temperatures would otherwise support higher rates, effectively conserving their carbohydrate reserves. This capacity for regulation is particularly important in environments where carbon gain during the day is limited by factors such as shading, drought, or nutrient deficiency. Plants that use crassulacean acid metabolism, or CAM, provide an interesting variation on the typical respiration pattern. CAM plants open their stomata at night to take in carbon dioxide, which they then store as organic acids in their vacuoles. During the day, the stomata close to conserve water, and the stored carbon dioxide is released internally and used for photosynthesis. This strategy allows CAM plants to photosynthesize while minimizing water loss in arid environments. However, it also means that their nighttime respiration rates are elevated compared to non-CAM plants because energy is required to drive the biochemical processes of carbon storage and acid synthesis. Despite this increased respiratory cost, the water savings often make CAM metabolism advantageous in dry habitats. The balance between photosynthesis and respiration determines a plant's net carbon gain. During the day, photosynthesis typically produces far more carbohydrates than respiration consumes, resulting in a net accumulation of biomass. At night, respiration continues, but photosynthesis does not, so there is a net loss of carbon as stored sugars are oxidized to provide energy. Over the course of a full day, the gain during daylight must exceed the loss at night for the plant to grow. This balance is sensitive to environmental conditions and can shift in response to changes in light, temperature, water availability, and nutrient supply. Respiration also plays a critical role in providing the building blocks and energy for growth. The intermediates of the citric acid cycle are precursors for the synthesis of amino acids, lipids, and other essential molecules. The ATP produced by respiration powers active transport processes that move ions and molecules across membranes, drives the synthesis of proteins and nucleic acids, and supports the mechanical work of cell expansion and division. Without a continuous supply of ATP from respiration, a plant would quickly exhaust its energy reserves and be unable to maintain its cellular functions. In addition to its role in energy metabolism, plant respiration is linked to the global carbon and oxygen cycles. Plants are often described as producers of oxygen and consumers of carbon dioxide, but this is only part of the story. While photosynthesis does release oxygen and fix carbon dioxide, respiration consumes oxygen and releases carbon dioxide, just as animal respiration does. The net effect when photosynthesis and respiration are both considered is that plants are net producers of oxygen and net consumers of carbon dioxide over the course of a day, but the balance is more nuanced than often portrayed. The oxygen in Earth's atmosphere is largely a result of billions of years of photosynthetic activity. Early in Earth's history, the atmosphere contained very little free oxygen. The evolution of photosynthesis in cyanobacteria, which are ancient photosynthetic microorganisms, gradually increased atmospheric oxygen levels over hundreds of millions of years, a transformation known as the Great Oxidation Event. This accumulation of oxygen made possible the evolution of aerobic respiration, which is far more efficient at extracting energy from organic molecules than anaerobic processes. Modern plants, which evolved much later, continue the work of oxygen production, maintaining the atmosphere in a state that supports diverse aerobic life. The interplay between photosynthesis and respiration in plants can be visualized as a continuous cycling of carbon and energy. Carbon dioxide from the air is captured and converted into sugars during photosynthesis. Some of those sugars are used immediately for respiration, being broken back down into carbon dioxide, which may be released back into the air or refixed in another cycle of photosynthesis. Some sugars are stored or used to build plant structures, representing a longer-term removal of carbon from the atmosphere. When plant tissues eventually die and decompose, the carbon they contain is oxidized by decomposers and returned to the atmosphere as carbon dioxide, completing the cycle. For a listener resting quietly, the concept of respiration as a mirror image of photosynthesis offers a complementary rhythm to contemplate. During the day, the leaf is a site of net synthesis, building molecules and releasing oxygen. At night, the leaf shifts to a state of net breakdown, consuming molecules and releasing carbon dioxide, though at a slower rate than the daytime synthesis. This daily oscillation, this breathing in and breathing out at the cellular level, continues throughout the life of the plant, as steady and reliable as the turning of the earth. The mitochondria themselves are descendants of ancient bacteria that entered into a symbiotic relationship with early eukaryotic cells, much as chloroplasts are descendants of photosynthetic bacteria. This endosymbiotic origin means that mitochondria retain some of their own DNA and ribosomes, distinct from those in the cell nucleus. They reproduce by division, independent of the cell cycle, and their membranes and biochemistry reflect their bacterial ancestry. The presence of both mitochondria and chloroplasts in plant cells is a testament to the power of symbiosis in evolution, with two once independent organisms now integrated into a single interdependent system. The quiet hum of respiration in plant cells, the steady consumption of oxygen and release of carbon dioxide, is largely invisible and inaudible, yet it is as essential to plant life as breathing is to animals. It provides the energy that drives growth, the power that moves water and nutrients, the fuel that supports reproduction and defense. It balances the exuberance of photosynthesis with the practical demands of cellular maintenance, ensuring that the energy captured from light is carefully allocated to the tasks that sustain life. In thinking about respiration, there is a gentle symmetry to appreciate. Oxygen released by photosynthesis is consumed by respiration. Carbon dioxide released by respiration is consumed by photosynthesis. The energy stored in sugars by photosynthesis is released by respiration. The cycle is closed, self-sustaining, and endlessly repeated. For a mind seeking rest, this image of balance and reciprocity of processes that complement and support each other can be deeply calming. It suggests a kind of equilibrium, a natural order in which opposing forces are harmonized and integrated into a coherent whole. Beneath the visible portion of every tree lies an equally complex and extensive system of roots spreading through the soil in search of water, nutrients, and stable anchorage. While the above-ground parts of a tree capture attention with their height and foliage, the roots perform essential work in darkness and silence, extracting resources from the earth and providing the foundation that supports the trunk and canopy. The architecture of root systems varies widely among species, but all share the basic functions of absorption, storage, and structural support. Contrary to a common misconception, tree roots do not typically extend as deep into the soil as the tree is tall. Most tree roots are concentrated in the top 60 to 90 centimeters of soil, where oxygen, moisture, and nutrients are most abundant. Some roots may penetrate deeper, particularly in search of water during dry periods, but the bulk of the root system spreads horizontally, often extending well beyond the drip line of the canopy. This lateral spread allows the tree to capture water and nutrients from a large area, increasing its access to resources and improving stability. The root system can be divided into different functional zones. Near the tips of roots are regions of active growth, where cells divide and elongate, pushing the root forward through the soil. Just behind the growing tip is the zone of maturation, where root hairs emerge. Root hairs are thin, delicate extensions of individual root cells that greatly increase the surface area available for absorption. A single root may produce thousands of root hairs, each only a few millimeters long, but collectively they can access a much larger volume of soil than the root itself. These root hairs are ephemeral, living for only a few days or weeks before being replaced by new ones as the root continues to grow. Water and dissolved minerals enter the root primarily through the root hairs, moving across cell membranes by osmosis and active transport. Osmosis is the movement of water from areas of higher water potential to areas of lower water potential, driven by differences in solute concentration. In the soil, water potential is generally higher than in the root cells, which contain dissolved sugars, salts, and other solutes, so water tends to move into the root. Once inside the root, water can travel through cell walls and intercellular spaces, or it can pass through the cells themselves, moving from the outer layers toward the central vascular tissue. At the center of the root is the vascular cylinder, which contains the xylem and phloem. Water and minerals that have entered the root must pass through a layer of cells called the endodermis to reach the xylem. The endodermis contains a waxy barrier known as the Casparian strip, which forces water and solutes to pass through the cell membranes rather than around them. This creates a checkpoint where the plant can regulate which substances enter the xylem and are transported to the rest of the plant. It is a quiet form of quality control, ensuring that the tree does not passively absorb every dissolved substance in the soil, but instead selectively takes up the nutrients it needs. Once water reaches the xylem, it begins its journey upward through the plant. Xylem tissue consists of dead, hollow cells arranged end to end to form continuous tubes that run from the roots to the leaves. The movement of water through xylem is driven primarily by transpiration, the evaporation of water from the leaves. As water evaporates from the leaf surfaces, it creates a negative pressure or tension in the xylem. This tension pulls water upward from the roots, much like sucking liquid through a straw. The cohesive properties of water molecules, which tend to stick together due to hydrogen bonding, allow this tension to be transmitted down the entire length of the xylem column without the water column breaking. Adhesion between water molecules and the walls of the xylem cells also helps support the column. This mechanism, known as the cohesion tension theory, explains how water can be lifted to great heights in tall trees without the need for any active pumping by the plant. The tallest trees, such as coast redwoods, can move water more than 100 meters from roots to crown, relying entirely on the physical properties of water and the driving force of transpiration. The process is continuous during the day when stomata are open and transpiration is occurring, and it slows at night when stomata close and evaporation decreases. Roots also interact extensively with soil microorganisms, including bacteria and fungi. One of the most important of these interactions is the symbiosis between plant roots and mycorrhizal fungi discussed earlier, which enhances nutrient uptake. Another critical partnership occurs in legumes and a few other plant families, which host nitrogen-fixing bacteria and specialized root structures called nodules. Nitrogen is an essential element for all living organisms, required for the synthesis of amino acids, proteins, and nucleic acids. Although nitrogen gas makes up about 78% of Earth's atmosphere, most organisms cannot use it in this form because the triple bond between the two nitrogen atoms is extremely strong and difficult to break. Certain bacteria, however, possess an enzyme called nitrogenase that can break this bond and convert nitrogen gas into ammonia, a form that plants can use. This process, known as nitrogen fixation, is energetically expensive, requiring significant amounts of ATP, but it provides access to an abundant resource. In the symbiosis between legumes and rhizobia bacteria, the plant provides the bacteria with sugars produced by photosynthesis, and the bacteria provide the plant with fixed nitrogen. The bacteria live inside the root nodules, where oxygen levels are carefully controlled. Nitrogenase is highly sensitive to oxygen and will not function if exposed to normal atmospheric concentrations, so the nodules contain a protein called leg hemoglobin, which binds oxygen and keeps it at low levels while still allowing some oxygen to reach the bacteria for respiration. Leg hemoglobin is similar in structure and function to the hemoglobin in animal blood, and it gives the inside of a healthy nodule a pinkish color. The fixed nitrogen produced in the nodules is transported throughout the plant and used to build proteins and other nitrogen-containing compounds. When legumes die and decompose, or when their roots are shed and decay, the nitrogen they contain is released into the soil, enriching it and benefiting neighboring plants. This is why legumes are often used in crop rotations and as cover crops. They improve soil fertility without the need for synthetic fertilizers. Roots also serve as storage organs for many plants. Carbohydrates produced by photosynthesis are transported from the leaves to the roots through the phloem and stored as starch in root cells. These reserves can be drawn upon during periods when photosynthesis is reduced, such as at night during winter dormancy, or when the plant is stressed. In some plants, such as carrots, beets, and sweet potatoes, the roots become highly specialized for storage, swelling with accumulated starch and sugars. In trees, stored carbohydrates in the roots support spring growth before the new leaves are fully functional, and they provide a buffer against environmental stresses such as drought or defoliation. The growth of roots is guided by environmental cues, a phenomenon known as tropism. Gravitropism causes roots to grow downward toward the pull of gravity while shoots grow upward. This directional growth is mediated by the redistribution of the hormone auxin in response to gravity. Specialized cells in the root tip contain dense starch-filled organelles called statoliths, which settle to the lower side of the cell under the influence of gravity. This settling triggers a signaling cascade that redirects auxin, which in turn influences cell elongation, causing the root to bend downward. Roots can also respond to other stimuli such as water and nutrients through a process called hydrotropism. Roots will preferentially grow toward areas of higher moisture, allowing the plant to access water even in heterogeneous soils. Similarly, roots may proliferate in soil patches that are rich in nutrients, optimizing resource capture. These growth responses are not conscious decisions, but are the result of differential cell growth and hormone signaling, finely tuned by evolution to maximize the plant's chances of survival. The physical act of root growth also involves considerable force. Roots must push aside soil particles, navigate around rocks, and sometimes penetrate compacted layers. The root tip is protected by a root cap, a cluster of cells that shields the delicate growing region and secretes mucilage, a slimy substance that lubricates the root's passage through the soil. As the root grows, cells in the cap are abraded and sloughed off, but they are continuously replaced by new cells produced just behind the tip. Over time, the root system of a tree becomes a vast and intricate network, with large structural roots providing anchorage and smaller feeder roots constantly exploring new regions of soil. The total length of roots, including all the fine roots and root hairs, can be many times the height of the tree, representing an enormous investment of resources. Yet this investment is essential because the roots are the tree's lifeline to water and nutrients, and without a healthy, extensive root system, the tree cannot sustain the metabolic demands of its leaves and growing tissues. For someone listening quietly, the image of roots spreading slowly through dark soil, extending delicate root hairs, forming partnerships with fungi and bacteria, and drawing up water molecule by molecule, offers a gentle and grounding mental picture. The roots work continuously without fanfare, performing tasks that are invisible from above but absolutely vital. They anchor the tree against wind and gravity. They mine the soil for nutrients. They store energy for future needs. And they communicate chemically with the hidden world of microorganisms. In their quiet persistence, they mirror the steady, unhurried processes that sustain all plant life. The movement of fluids within a plant is a constant, invisible process that sustains every living cell from root tip to leaf edge. Two distinct vascular systems perform this work. Xylem, which transports water and dissolved minerals upward from the roots, and phloem, which distributes sugars and other organic compounds throughout the plant. Together, these systems form a circulatory network that, while fundamentally different from the blood vessels of animals, serves a similarly vital role in maintaining the internal environment and supporting growth. Xylem tissue is composed of specialized cells that, when mature, are no longer alive. In flowering plants, the primary water-conducting cells are called vessel elements and tracheids. Vessel elements are short, wide cells that stack end to end, with their end walls either entirely dissolved or perforated to form large openings. This creates continuous tubes or vessels that can extend for many meters through the plant. Tracheids are narrower and more elongated with tapered ends that overlap. Water moves from one tracheid to the next through small pits in the cell walls, which are areas where the secondary wall is absent and only the thin primary wall remains. Both vessel elements and tracheids have thick lignified secondary walls that provide structural support and prevent the cells from collapsing under the negative pressure generated by transpiration. Lignin is a complex polymer that adds rigidity to cell walls and is one of the reasons wood is so strong. The patterns of lignin deposition in xylem cells can be seen under a microscope as rings, spirals, or networks, each pattern providing a different balance between strength and flexibility. Water enters the xylem in the roots and moves upward through the stem and into the leaves, driven primarily by the pull of transpiration. Transpiration is the loss of water vapor from the plant, occurring mainly through stomata, the tiny pores on leaf surfaces. As water molecules evaporate from the moist surfaces of the cells inside the leaf and diffuse out through the stomata, they create a tension in the water column within the xylem. This tension is transmitted downward through the interconnected water molecules, pulling more water up from the roots. The cohesion tension theory explains how this process can lift water to great heights. Water molecules are cohesive, meaning they stick to each other through hydrogen bonds. This cohesion allows the water in the xylem to behave somewhat like a solid column that can be pulled upward without breaking apart. Water molecules also adhere to the walls of the xylem cells, which helps support the column and prevents it from pulling away from the cell walls under tension. Together, cohesion and adhesion make it possible for water to move from roots to leaves and even the tallest trees without any active pumping by the plant. The rate of water movement through xylem can be quite rapid during periods of high transpiration. On a hot, sunny day, a large tree may transport hundreds of liters of water from soil to atmosphere. This flow is not constant, however. It varies with environmental conditions such as light intensity, temperature, humidity, and wind, all of which affect the rate of transpiration. At night, when stomata close and transpiration slows, the flow of water through xylem decreases markedly. The plant's internal water status changes over the course of a day, with water content typically highest in the early morning after a night of reduced transpiration and lowest in the late afternoon after hours of water loss. In very tall trees, the challenge of transporting water to the top of the canopy is immense. Coast redwoods and giant sequoias, which can exceed 90 meters in height, rely on several adaptations to move water efficiently. One factor is the presence of fog in their native habitats. Fog can be absorbed directly by leaves, reducing the need for water to be drawn all the way from the roots and easing the tension in the xylem. Additionally, the xylem in very tall trees must be highly resistant to cavitation, a phenomenon where the water column breaks and forms an air bubble, or embolism that blocks further water transport. Trees have evolved xylem structures that minimize the risk of cavitation and mechanisms to repair or bypass embolisms when they do occur. Phloem, the second major vascular tissue, transports sugars and other organic compounds from sites of production, typically the leaves, to sites of use or storage, such as roots, fruits, and growing tissues. Unlike xylem, phloem is composed of living cells. The main conducting cells in phloem are called sieve tube elements, which are arranged end-to-end to form sieve tubes. The end walls of sieve tube elements, called sieve plates, are perforated with pores that allow the flow of fluid from one cell to the next. Sieve tube elements are unusual in that they lack nuclei and many other organelles at maturity, having expelled or broken down these structures to maximize the space available for fluid flow. However, they remain alive and metabolically active, supported by adjacent companion cells. Companion cells are connected to sieve tube elements by numerous plasmodesmata, tiny channels through the cell walls that allow direct cytoplasmic connections. The companion cells provide the sieve tube elements with proteins, energy, and other materials necessary for their function. The mechanism of phloem transport is more complex than that of xylem and is described by the pressure flow hypothesis. According to this model, sugars produced in the leaves by photosynthesis are actively loaded into the sieve tubes either directly from the photosynthetic cells or via companion cells. The high concentration of sugar in the sieve tubes causes water to enter by osmosis from the surrounding xylem, increasing the pressure inside the phloem. At the other end of the plant, in roots or other sink tissues, sugars are actively unloaded from the sieve tubes and used for growth, storage, or respiration. This removal of sugar lowers the solute concentration, causing water to leave the phloem by osmosis and return to the xylem. The result is a pressure gradient along the phloem, with high pressure at the source, where sugars are loaded, and low pressure at the sink, where sugars are unloaded. This gradient drives the flow of fluid from source to sink, carrying dissolved sugars along with it. The process is continuous as long as there is a difference in sugar concentration between source and sink, and it does not require the expenditure of energy for the bulk flow itself, though energy is needed for the active loading and unloading of sugars. Phloem can transport materials in multiple directions depending on where the sources and sinks are located. For example, during the growing season, a mature leaf acts as a source, exporting sugars downward to the roots and upward to developing fruits or young leaves. In spring, when new leaves are growing and not yet photosynthetically active, stored sugars in the roots become the source, and the phloem transports them upward to support new growth. This bidirectional capacity makes phloem a highly flexible distribution system. The compounds transported in phloem include not only sugars, primarily sucrose, but also amino acids, hormones, signaling molecules, and even small RNAs that may play roles in gene regulation and communication between different parts of the plant. The phloem sap is thus a rich and complex fluid carrying information and resources to coordinate growth and development across the entire organism. The structure of vascular tissues in stems and roots is carefully organized. In most stems, xylem and phloem are arranged in vascular bundles, with xylem typically on the inside and phloem on the outside. In woody plants, a layer of dividing cells called the vascular cambium lies between the xylem and phloem. The cambium produces new xylem cells toward the inside and new phloem cells toward the outside, causing the stem to increase in diameter over time. This secondary growth is what produces wood, which is essentially accumulated layers of xylem. The annual addition of xylem in temperate climates creates the growth rings visible in a cross-section of a tree trunk. The outer layers of phloem in woody plants are eventually crushed and incorporated into the bark as new phloem is produced. Bark serves as a protective layer, shielding the living tissues of the stem from physical damage, temperature extremes, and pathogens. It is composed of dead phloem, cork cells, and other tissues, and it can be quite thick in some species, providing insulation and fire resistance. The continuous operation of xylem and phloem, moving water upward and sugars in multiple directions, can be visualized as a series of slow, steady streams flowing through the plant. Water rises through the xylem, pulled by the gentle but relentless force of evaporation at the leaves. Sugars glide through the phloem, pushed by pressure gradients created by loading and unloading. Both processes are passive in the sense that the bulk flow itself does not require energy input, though the underlying mechanisms of transpiration, sugar loading, and sugar unloading do involve active processes. For a mind seeking calm, the image of these internal rivers offers a peaceful focal point. The flow is invisible, silent, and continuous. Water molecules travel upward, one after another, cohering to each other and adhering to the xylem walls, responding to the pull from above. Sugar molecules dissolve in phloem sap, carried along by the flow of water, moving from leaf to root or from root to bud, distributing the energy captured by photosynthesis. The system is elegant in its simplicity and efficiency, a testament to the power of evolution to shape functional structures from basic physical principles. Although plants lack muscles and nervous systems, they are capable of a surprising variety of movements, both slow and in some cases remarkably fast. These movements are driven by changes in cell turgor pressure, differential growth rates, or specialized structures that can generate mechanical force. Plant movements can be categorized broadly into tropisms, which are directional growth responses to environmental stimuli, and nastic movements, which are non-directional responses that occur regardless of the direction of the stimulus. Phototropism is the growth of a plant toward or away from a light source. Shoots typically exhibit positive phototropism, bending toward light, while roots may show negative phototropism, growing away from light. The mechanism behind phototropism involves the plant hormone auxin, which is produced in the shoot tip and transported downward. When light strikes one side of the shoot more than the other, photoreceptor proteins in the cells detect the difference and cause auxin to accumulate on the shaded side. Auxin stimulates cell elongation, so the cells on the shaded side grow longer than those on the illuminated side, causing the shoot to bend toward the light. This bending is gradual, occurring over hours or days, and it allows the plant to optimize its light capture by adjusting the position of its leaves and stem. The response is highly sensitive. Even diffuse light from a cloudy sky can induce phototropic bending. Over the course of a plant's life, phototropism helps ensure that leaves are positioned to receive maximum sunlight, which is critical for photosynthesis. Gravitropism, also called geotropism, is the growth response to gravity. Roots exhibit positive gravitropism, growing downward, while shoots exhibit negative gravitropism, growing upward. The sensing of gravity occurs in specialized cells that contain statoliths, dense, starch-filled organelles that settle to the bottom of the cell under the influence of gravity. This settling triggers a redistribution of auxin, which then affects the rate of cell elongation on different sides of the root or shoot, causing it to bend. In roots, auxin accumulation on the lower side inhibits cell elongation, so the upper side grows faster and the root bends downward. In shoots, auxin on the lower side promotes cell elongation, so the lower side grows faster and the shoot bends upward. This differential response to the same hormone in different tissues is an elegant example of how plants fine-tune their growth through context-dependent signaling. Thigmotropism is the growth response to touch or physical contact. Climbing plants such as vines and ivy exhibit positive thigmotropism, wrapping around supports when they make contact. The tendrils of pea plants and grapevines are particularly sensitive to touch and will begin to coil around an object within minutes of contact. This response involves changes in cell growth rates on opposite sides of the tendril, with cells on the side away from the stimulus elongating more rapidly, causing the tendril to curve around the support. Over time, the tendril tightens and strengthens, securing the plant to its support and allowing it to climb. Nastic movements, unlike tropisms, do not depend on the direction of the stimulus. They are typically faster than tropisms and are driven by reversible changes in turgor pressure rather than by growth. One of the most well-known examples is the closure of the leaflets of the sensitive plant, Mimosa pudica, when touched. Within seconds of being disturbed, the leaflets fold together and the petiole droops. This rapid movement is caused by the sudden loss of turgor in specialized cells at the base of each leaflet and petiole, called pulvini. Water and ions rapidly exit these cells, causing them to lose rigidity and the leaf to collapse. The movement is thought to deter herbivores or reduce water loss by decreasing the exposed leaf area. After a period of time, the cells regain turgor and the leaf reopens. Nyctinasty is the nastic movement of leaves in response to the daily cycle of light and darkness. Many plants, including beans and clovers, fold their leaves at night and open them during the day. This movement is controlled by the circadian clock and involves changes in turgor pressure in the pulvini. The adaptive significance of nyctinasty is not entirely clear, but it may reduce heat loss at night, minimize exposure to nocturnal herbivores, or reduce water loss through transpiration. Carnivorous plants have evolved some of the most dramatic and rapid movements in the plant kingdom. The Venus flytrap, Dionea muscipula, can snap its leaves shut in a fraction of a second when trigger hairs inside the trap are touched. This remarkable speed is achieved through a combination of elastic energy storage and rapid changes in cell shape. The trap leaves are held open in a state of tension, with cells on the outer surface curved outward. When mechanical stimulation of the trigger hairs exceeds a threshold, electrical signals are generated and spread across the leaf. These signals trigger a rapid movement of water and ions out of certain cells, causing a sudden change in curvature that releases the stored elastic energy and snaps the trap shut. The Venus flytrap also demonstrates a form of counting and memory. A single touch of a trigger hair is not sufficient to close the trap. The hairs must be touched at least twice within a short time window, typically about 20 seconds. This prevents the trap from closing in response to random stimuli such as raindrops or falling debris, conserving the plant's energy for actual prey. Once the trap is closed around an insect, continued stimulation of the trigger hairs signals the presence of prey and initiates the secretion of digestive enzymes. If no further stimulation occurs, indicating a false alarm, the trap will reopen after a day or so. Pitcher plants use a different trapping strategy, creating tubular or urn-shaped leaves that collect rainwater and digestive fluids. Insects are attracted to the pitcher by nectar or bright colors and then slip into the fluid-filled chamber where they drown and are digested. Some pitcher plants have evolved slippery surfaces or downward-pointing hairs that prevent insects from escaping once they have entered. One species, Nepenthes gracilis, has been found to use a rapid, spring-loaded trapping mechanism on the lid of the pitcher, which can flip inward when an insect lands on it, catapulting the insect into the pitcher. This movement occurs in milliseconds and relies on the sudden release of stored elastic energy, similar to the mechanism in the Venus flytrap. The ability of plants to move, whether slowly through growth or rapidly through turgor changes, challenges the common perception of plants as static organisms. While their movements are different from those of animals, they are no less sophisticated, involving complex sensory systems, signal transduction pathways, and mechanical structures. Plants can sense light, gravity, touch, chemicals, temperature, and even the presence of other organisms, and they respond to these stimuli in ways that enhance their survival and reproduction. The hormones that regulate plant movements are diverse and interact in complex ways. Auxin, already mentioned in the context of tropisms, is just one of several major plant hormones. Gibberellins promote stem elongation and seed germination. Cytokinins stimulate cell division and delay aging in leaves. Ethylene is a gaseous hormone involved in fruit ripening, leaf abscission, and stress responses. Abscisic acid regulates stomatal closure and promotes dormancy. These hormones are produced in various parts of the plant and transported to target tissues, where they bind to receptors and trigger changes in gene expression or cellular activity. The coordination of plant movements and growth through hormones allows the plant to integrate information from multiple sources and respond in a coherent manner. For example, a plant experiencing drought might produce abscisic acid, which closes stomata to conserve water while simultaneously redirecting auxin to promote root growth and increase water uptake. The plant is constantly balancing competing demands and adjusting its physiology to match its environment. For a listener resting quietly, the concept of plants moving in slow motion, bending toward light, curling around supports, or folding their leaves at dusk provides a gentle and rhythmic image. These movements are not random but are carefully regulated responses to the world around the plant. They unfold over minutes, hours, or days with a patience and consistency that mirrors the slow turning of the seasons. Even the rapid snap of a Venus flytrap, which happens in milliseconds, is the culmination of slow processes of growth, sensing, and energy storage. The image of a tendril slowly coiling around a stick, or a shoot gradually bending toward a window, or a leaf folding as night falls can serve as a calming focal point for the mind. These are small, quiet events, easily overlooked, yet they reveal the dynamic nature of plant life. They remind us that even in the apparent stillness of a forest or garden, there is constant adjustment, constant response, constant movement, all occurring at a pace that invites slow, restful observation. On the surface of nearly every leaf, invisible to the naked eye, are thousands of tiny pores called stomata. These microscopic openings serve as the gateways for gas exchange between the plant and the atmosphere, allowing carbon dioxide to enter for photosynthesis and oxygen to exit as a byproduct. At the same time, water vapor escapes through stomata during transpiration, creating the tension that pulls water upward through the xylem. The regulation of stomatal opening and closing is one of the most critical processes in plant physiology, balancing the need for carbon dioxide intake against the need to conserve water. Each stoma, the singular form of stomata, consists of a pore surrounded by a pair of specialized cells called guard cells. Guard cells are shaped like kidney beans or crescent moons, with their curved sides facing each other and forming the pore between them. The cell walls of guard cells are unevenly thickened, with the wall adjacent to the pore being thicker and less flexible than the outer wall. This structural asymmetry is key to how stomata open and close. When guard cells take up water and become turgid, they swell. Because of the uneven thickness of their walls, the cells bow outward, pulling the pore open. When guard cells lose water and become flaccid, they relax, and the pore closes. This simple mechanical principle, driven by changes in turgor pressure, allows the plant to rapidly adjust the size of the stomatal opening in response to environmental conditions and internal signals. The movement of water into and out of guard cells is controlled by the movement of ions, particularly potassium ions, across the cell membrane. When stomata need to open, such as in the morning when light becomes available for photosynthesis, the guard cells actively transport potassium ions into their cytoplasm from surrounding cells. This increases the solute concentration inside the guard cells, causing water to enter by osmosis. The resulting increase in turgor pressure opens the pore. When stomata need to close, potassium ions are pumped out of the guard cells. Water follows by osmosis, turgor pressure decreases, and the pore closes. The triggers for stomatal opening and closing are numerous and interconnected. Light is a major signal. Blue light in particular is detected by photoreceptor proteins in the guard cells, which activate signaling pathways that lead to potassium uptake and stomatal opening. This ensures that stomata are open during the day when photosynthesis can occur. At night, in the absence of light, stomata typically close, reducing water loss when carbon dioxide is not needed for photosynthesis. Carbon dioxide concentration inside the leaf also affects stomatal behavior. When carbon dioxide levels are low, indicating that photosynthesis is consuming carbon dioxide faster than it is entering the leaf, stomata open wider to allow more carbon dioxide in. Conversely, when carbon dioxide levels are high, stomata close partially to prevent excessive water loss. This feedback loop helps the plant balance carbon gain and water loss. Water availability is another critical factor. When a plant experiences water stress, such as during drought, it produces the hormone abscisic acid in its roots and transports it to the leaves. Abscisic acid binds to receptors in the guard cells and triggers a cascade of events that lead to the loss of potassium ions and water, causing stomata to close. This response helps the plant conserve water during times of scarcity, though it comes at the cost of reduced photosynthesis and slower growth. Humidity also plays a role. In dry air, the rate of water loss through open stomata is higher than in humid air. Plants can sense the humidity of the surrounding air, possibly through the rate of water loss itself, or through specialized cells that detect changes in the water status of the leaf, and they adjust stomatal aperture accordingly. On a hot, dry day, stomata may close partially even in bright light to prevent excessive dehydration. The daily rhythm of stomatal opening and closing is also influenced by the plant's circadian clock. Even in constant environmental conditions, many plants will continue to open and close their stomata on a roughly 24-hour cycle, anticipating the normal pattern of day and night. This anticipatory regulation allows the plant to prepare for photosynthesis before dawn and to conserve water before dusk, improving overall efficiency. The density and distribution of stomata vary widely among plant species and even among different leaves on the same plant. Plants adapted to dry environments often have fewer stomata or have them sunken into pits in the leaf surface, reducing exposure to dry air and minimizing water loss. Aquatic plants, which have abundant water, may have stomata only on the upper surface of floating leaves, where gas exchange with the atmosphere is necessary. plants in shaded environments may have larger or more numerous stomata to maximize carbon dioxide uptake under low light conditions. The structure of the leaf epidermis surrounding the stomata also contributes to gas exchange regulation. The epidermis is typically covered by a waxy cuticle that is impermeable to water and gases, preventing uncontrolled water loss. Stomata are the only openings through this barrier, making their regulation essential for controlling the plant's water budget. Beneath each stoma, inside the leaf, is an airspace surrounded by loosely packed mesophyll cells. This airspace allows carbon dioxide to diffuse from the atmosphere into the interior of the leaf, where it can reach the chloroplasts and the mesophyll cells. Similarly, oxygen produced by photosynthesis diffuses from the mesophyll into the airspace and exits through the stomata. The efficiency of this gas exchange system depends on maintaining a balance between the surface area available for diffusion and the resistance to water loss. Leaves are typically thin and flat, maximizing surface area relative to volume and minimizing the distance gases must diffuse to reach the photosynthetic cells. The spongy structure of the mesophyll, with its air spaces and interconnected cells, provides a large internal surface area for gas exchange. The regulation of stomatal aperture allows the plant to fine-tune the rate of gas exchange, opening wide when conditions are favorable and closing partially when water is scarce or carbon dioxide demand is low. Transpiration, the loss of water through stomata, serves several functions beyond simply being a consequence of gas exchange. It drives the upward movement of water and nutrients through the xylem, as discussed earlier. It also cools the leaf through evaporative cooling, much as sweating cools the human body. On a hot day, a leaf can be several degrees cooler than the surrounding air due to transpiration, which helps prevent heat damage to the photosynthetic machinery. However, transpiration also represents a significant cost. A large tree can transpire hundreds of liters of water per day, and this water must be continuously replaced from the soil. If water availability is limited, the plant must reduce transpiration to avoid desiccation, but this also reduces carbon dioxide uptake and photosynthesis. This trade-off between carbon gain and water loss is a central challenge in plant life, and the regulation of stomata is the primary means by which plants manage it. The study of stomata has revealed their remarkable sensitivity and responsiveness. Stomata can respond to changes in light, carbon dioxide, humidity, and hormones within minutes. They integrate multiple signals and adjust their aperture to optimize the plant's performance under current conditions. This dynamic regulation is essential for survival in variable environments and is a key factor in determining a plant's productivity and water use efficiency. For someone listening in a state of relaxation, the image of thousands of tiny pores opening and closing across the surface of a leaf, each one responding to the same signals in coordinated fashion, offers a meditative visual. The guard cells swell and relax. The pores widen and narrow and gases diffuse in and out, all in a slow, rhythmic dance regulated by light, water, and internal clocks. It is a process that is continuous and largely invisible, yet it sustains the life of the plant and contributes to the oxygen in the air we breathe. The quiet efficiency of this system, the precision with which it balances competing needs, can inspire a sense of calm and appreciation for the ordered complexity of living systems. Among the most remarkable organisms on Earth are trees that have lived for thousands of years, silently witnessing the passage of centuries and recording in their wood the fluctuations of climate, the rhythms of wet and dry seasons, and the slow march of environmental change. These ancient trees are not merely survivors, they are living archives, their growth rings preserving information that scientists can use to reconstruct past climates and understand long-term ecological patterns. The oldest known individual trees are bristlecone pines, Pinus longaiva, which grow in the high mountains of the western United States. One bristlecone pine, known as Methuselah, has been dated to over 4,800 years old, making it one of the oldest living organisms on the planet. Its exact location is kept secret to protect it from vandalism. These trees grow in harsh, arid environments with poor soils and extreme temperature fluctuations, conditions that slow their growth but also limit competition and reduce the risk of disease and fire. Bristlecone pines grow very slowly, sometimes adding only a fraction of a millimeter to their diameter in a year. This slow growth contributes to the density and durability of their wood, which is highly resistant to decay. Even after a bristlecone pine dies, its wood can remain intact for thousands of additional years, weathering in place and preserving a record of its growth. By cross-dating the rings of living trees with those of dead trees and ancient timbers, scientists have constructed continuous chronologies extending back more than 11,000 years, far beyond the lifespan of any single tree. The study of tree rings is called dendrochronology, and it relies on the fact that trees in seasonal climates produce a distinct ring each year. During the growing season, usually spring and early summer, trees produce large, thin-walled cells that form light-colored early wood. Later in the season, growth slows and the cells produced are smaller and have thicker walls, forming darker late wood. The transition from the late wood of one year to the early wood of the next creates a visible boundary, the ring, which can be counted to determine the age of the tree. The width of each ring reflects the environmental conditions during that year. A wide ring indicates favorable conditions such as ample moisture and moderate temperatures which promote rapid growth. A narrow ring indicates stress such as drought, extreme cold, or competition for resources which limit growth. By measuring the width of rings across many trees from the same region, scientists can identify patterns that reflect regional climate variations. Years of drought, for example, will produce narrow rings in many trees, creating a distinctive pattern that can be matched across samples. These patterns allow for cross-dating, the process of matching ring width sequences from different trees to establish the calendar year in which each ring was formed. Cross-dating is the foundation of dendrochronology and enables the construction of long chronologies that extend beyond the lifespan of individual trees. By overlapping the ring patterns of living trees, dead trees, and ancient timbers, scientists can build a continuous record of environmental conditions stretching back millennia. Tree rings have been used to reconstruct past climates with remarkable precision. They provide annual resolution, meaning that scientists can identify specific years of drought, cold, or favorable conditions. This level of detail is rare in paleoclimate records and makes tree rings particularly valuable for understanding climate variability and change. Tree ring records have been used to study phenomena such as the medieval warm period, the Little Ice Age, and the timing and severity of historical droughts. In addition to ring width, other properties of the wood can provide climatic information. The density of the wood, the isotopic composition of the cellulose, and the chemical content of the rings can all reflect environmental conditions. For example, the ratio of oxygen isotopes in tree rings is influenced by temperature and precipitation, providing an independent line of evidence for climate reconstruction. By combining multiple types of data from tree rings, scientists can develop a more complete picture of past climates. Tree rings have also been used to date archaeological sites and historical events. By matching the ring patterns of timbers used in ancient buildings or artifacts with established tree ring chronologies, researchers can determine the year in which the wood was cut, providing precise dates for construction or use. This technique has been applied to pueblos in the southwestern United States, Viking ships, and historic buildings in Europe, among many other examples. Beyond bristlecone pines, other trees achieve great ages. Giant sequoias, Sequoia dendron giganteum, can live for over 3,000 years. These massive trees, found in scattered groves in the Sierra Nevada of California, are among the largest living organisms by volume. A single giant sequoia can contain enough wood to build dozens of houses, and its trunk can exceed 10 meters in diameter. The bark of a giant sequoia can be more than half a meter thick, providing insulation against fire and physical damage. Coast redwoods, Sequoia sempervirens, are the tallest trees on Earth, with the tallest known individual named Hyperion measuring over 115 meters in height. These trees grow in the fog belt of coastal California and southern Oregon, where frequent fog provides moisture that supplements rainfall and allows the trees to reach such extreme heights. The ability of redwoods to absorb water directly from fog through their leaves reduces the stress on the xylem and allows them to transport water to the top of their towering canopies. The longevity of these trees is a testament to their resilience and the stability of their environments. They have evolved mechanisms to resist fire, disease, and physical damage. Their thick bark, high tannin content, and ability to re-sprout from the base after injury all contribute to their survival over centuries. They also benefit from living in environments where competition is limited and disturbances such as logging or land clearing have been minimal, at least until recent times. The existence of trees that predate human civilizations, that were already ancient when the pyramids were built, invites a contemplative perspective on time. A bristlecone pine that germinated nearly 5,000 years ago has lived through the entire span of recorded human history. It has weathered countless storms, droughts, and cold snaps. It has grown imperceptibly slowly, adding ring after ring, year after year, in a rhythm that has continued for millennia. For a listener seeking calm, the image of an ancient tree standing alone on a mountainside, its twisted trunk shaped by wind and time, its roots gripping the rocky soil, offers a profound sense of continuity and endurance. The tree does not hurry. It does not measure its life in days or years, but in centuries. Each ring in its wood is a marker of time, a record of a year that came and went, a year of growth, however slight. The accumulation of these rings, one atop the other, builds a structure that can stand for thousands of years, a living monument to patience and persistence. The study of these ancient trees and their rings connects the present to the distant past. It allows scientists to see patterns and trends that span far longer than a human lifetime, providing context for current climate change and environmental shifts. It also offers a reminder that the natural world operates on multiple timescales, from the rapid movements of a Venus flytrap to the slow, steady growth of a bristlecone pine. Both are expressions of life. Both are the result of evolutionary processes, and both invite a sense of wonder at the diversity and adaptability of living organisms. The rings themselves, visible in a cross-section of wood, form a pattern of concentric circles, each one slightly different in width and texture. Running a finger across these rings from the center to the bark is to trace a path through time, moving outward from the tree's youth to its present age. The innermost rings were formed when the tree was a seedling, vulnerable and small. The outermost rings were formed recently when the tree was already ancient, its growth slowed but still continuing. The quiet, repetitive pattern of the rings, the gentle variation in their width, can serve as a calming visual, a reminder of the steady passage of time and the endurance of life. The tallest trees on Earth are coast redwoods, Sequoia sempervirens, which inhabit a narrow strip of coastal land in Northern California and Southern Oregon. These giants can exceed 115 meters in height, with the current record holder, a tree named Hyperion, standing at approximately 116 meters tall. To put this in perspective, a coast redwood at this height is taller than the Statue of Liberty from base to torch. The ability of these trees to grow so tall represents a remarkable feat of structural engineering and physiological adaptation. Several factors limit the maximum height that a tree can reach. One of the primary constraints is the challenge of transporting water from the roots to the topmost leaves. As height increases, the tension required to pull water upward through the xylem increases, and the risk of cavitation, where the water column breaks and forms air bubbles, also increases. At extreme heights, the negative pressure in the xylem approaches the physical limits of what water can withstand before the column fails. Coast redwoods have evolved several adaptations that help them overcome this challenge. One of the most important is their ability to absorb water directly from fog through their leaves. The coastal regions where redwoods grow experience frequent fog, especially during the summer months when rainfall is scarce. As fog passes through the canopy, water condenses on the needles and branches and can be absorbed through the leaf surfaces or drip to the ground, where it is taken up by the roots. This fog drip can contribute a significant amount of water to the tree, reducing the demand on the xylem to transport water from the soil and easing the tension in the water column. Another adaptation is the structure of the xylem itself. Redwoods have xylem cells that are highly resistant to cavitation, with small pore sizes and thick walls that help maintain the integrity of the water column even under high tension. If cavitation does occur, redwoods have mechanisms to isolate the affected vessels and route water through alternative pathways, minimizing the impact on overall water transport. The structural support required to hold up a tree of such height is also formidable. The trunk of a coast redwood must be strong enough to support the weight of its branches and foliage, and it must be flexible enough to withstand the forces of wind. Redwood wood is dense and strong, with high tensile and compressive strength, allowing it to bear the enormous loads involved. The tapered shape of the trunk, widest at the base and gradually narrowing toward the top, distributes the weight efficiently and reduces bending stresses. The root system of a redwood is surprisingly shallow for such a tall tree, typically extending only 3 to 4 meters deep, but spreading widely, often 50 to 80 meters from the trunk. Redwoods often grow in groves, and the roots of neighboring trees intertwine, creating a network that provides mutual support and stability. This interconnected root system helps anchor the trees against wind throw and distributes the load across multiple individuals. The canopy of a tall redwood is a complex, layered environment. The uppermost branches receive full sunlight and are exposed to wind and temperature extremes. Lower in the canopy, light levels decrease and the microclimate becomes more stable, with higher humidity and lower temperatures. Different species of plants, lichens, mosses, and even other trees can grow as epiphytes in the branches of redwoods, creating a vertical ecosystem with distinct zones. Forest canopies in general are structured into layers, each with characteristic light, temperature, and moisture conditions. The emergent layer consists of the tallest trees which rise above the main canopy. The canopy layer forms a continuous cover of foliage that intercepts most of the sunlight. Below this is the understory where smaller trees and shrubs grow in reduced light. The forest floor, which receives only a small fraction of the light that strikes the canopy, is home to shade-tolerant plants, fungi, and decomposers. The vertical stratification of the forest creates a diversity of habitats and microclimates. Temperature can vary by several degrees between the canopy and the forest floor. Humidity is typically higher near the ground and lower in the canopy where air circulation is greater. Wind speeds are highest in the emergent layer and decrease sharply within the canopy and understory. These gradients influence the distribution of species and the ecological interactions within the forest. The height of a tree also affects its photosynthetic efficiency. Leaves at the top of the canopy receive abundant light and can photosynthesize at high rates, but they also experience greater water stress and higher temperatures, which can inhibit photosynthesis. Leaves lower in the canopy receive less light but are in a more stable environment with lower water stress. The tree must balance the allocation of resources between upper and lower leaves, ensuring that both contribute to overall carbon gain. There is evidence that height growth in trees eventually slows and stops as they approach their maximum height. This leveling off is thought to be due to the increasing difficulty of transporting water to the top of the tree and the declining efficiency of photosynthesis in the uppermost leaves. At extreme heights, the leaves at the top may be operating near their physiological limits, with high water stress and reduced photosynthetic rates. Further height growth would provide diminishing returns, and the tree instead allocates resources to widening its trunk, expanding its root system, or producing seeds. The age of the tallest redwoods is also impressive, though not as extreme as that of bristlecone pines. Coast redwoods can live for over 2,000 years, with some individuals dating back to the time of ancient Rome. Their longevity is supported by their resistance to fire, pests, and disease, as well as their ability to resprout from the base if the main trunk is damaged. Redwood bark is thick, fibrous, and rich in tannins, which makes it resistant to fire and decay. Many old-growth redwoods bear fire scars from past blazes, yet they continue to grow and thrive. The experience of standing at the base of a coast redwood and looking upward is one of profound scale. The trunk rises straight and tall, disappearing into the canopy high above. The lower branches may be tens of meters off the ground, and the crown, where the foliage is concentrated, is often hidden from view. The sheer vertical distance is difficult to grasp, and the silence and stillness of the forest add to the sense of awe. For a listener resting in the quiet, the image of a redwood stretching upward, its top lost in fog, its roots spreading wide beneath the forest floor, offers a sense of stability and grandeur. The tree has grown slowly year after year, adding height and girth in a patient accumulation of wood and foliage. It has weathered storms and droughts, fires and floods, and it continues to stand, a monument to the possibilities of growth and endurance. The quiet verticality of the redwood, the way it reaches steadily upward without haste or drama, can serve as a calming mental image, a reminder that growth is a process, not an event, and that the tallest structures are built slowly, over long periods of time. The fog that drifts through the redwood canopy, condensing on needles and dripping to the ground, is itself a gentle, repetitive phenomenon. Each droplet forms slowly, grows and falls, adding a tiny increment of moisture to the forest. Over the course of a summer, this fog drip can deliver as much water as rainfall in other seasons, sustaining the trees and the understory during the dry months. The image of fog moving silently through the branches, of water condensing and dripping, of trees drawing moisture from the air is soothing in its slowness and constancy. It is a process that happens every day, unnoticed and unheralded, yet essential to the life of the forest. A seed is a package of potential, a small structure that contains an embryonic plant, a supply of stored nutrients, and a protective coat, all waiting for the right conditions to begin growth. Seeds are among the most remarkable adaptations in the plant kingdom, allowing plants to survive harsh conditions, disperse to new locations, and delay germination until the environment is favorable. The life of a seed is defined by patience, by the ability to wait, sometimes for years or even decades, until the moment is right to grow. Seed dormancy is the state in which a viable seed will not germinate even when provided with adequate water, oxygen, and suitable temperatures. Dormancy is an adaptive strategy that prevents germination at inappropriate times, such as in late autumn when a seedling would face freezing temperatures before it could establish itself. By remaining dormant through unfavorable periods, seeds increase the likelihood that germination will occur when conditions support survival and growth. There are several types of dormancy, each with different mechanisms and triggers. Physiological dormancy is the most common type and involves biochemical processes within the seed that inhibit germination. The embryo may produce hormones, particularly abscisic acid, that suppress growth and keep the seed in a resting state. Germination can be triggered by changes in hormone balance, such as a decrease in abscisic acid and an increase in gibberellins, which promote cell division and elongation. Physical dormancy occurs when the seed coat is impermeable to water or gases, preventing the embryo from taking up the water needed to begin growth. Seeds with physical dormancy often require scarification, the mechanical or chemical weakening of the seed coat before they can germinate. In nature, scarification can occur through the action of fire, the digestive processes of animals that eat the seeds, or the abrasive effects of water and soil. Once the seed coat is breached, water can enter and germination can proceed. Morphological dormancy occurs when the embryo is not fully developed at the time of seed dispersal. The seed must undergo a period of after-ripening, during which the embryo continues to develop before it is capable of germinating. This type of dormancy is common in certain plant families and ensures that seeds do not germinate until the embryo is mature enough to sustain growth. Combinational dormancy involves a combination of physical and physiological mechanisms. Seeds with this type of dormancy require multiple conditions to be met before germination can occur, such as both scarification of the seed coat and exposure to specific temperature cycles. This complexity ensures that germination happens only when a precise set of environmental cues indicates that conditions are optimal. The breaking of dormancy often requires exposure to specific environmental signals. For many temperate species, seeds must experience a period of cold stratification, meaning they need to be exposed to cold, moist conditions for several weeks or months. This requirement ensures that seeds do not germinate in autumn, only to have the seedlings killed by winter frost. Instead, the seeds remain dormant through the winter, and the cold period itself serves as a signal that winter has passed and spring, with its favorable growing conditions, is approaching. Light is another important signal for some seeds. Certain small-seeded species require exposure to light to germinate, ensuring that they only sprout when they are near the soil surface where the seedling will have access to light for photosynthesis. Seeds buried deep in the soil where light cannot penetrate remain dormant until disturbance brings them to the surface. This can result in seed banks, populations of dormant seeds in the soil that can persist for years, waiting for the right conditions to germinate. Fire is a germination trigger for some species, particularly those adapted to fire-prone environments. The heat from a fire can crack hard seed coats, allowing water to enter, or it can stimulate the release of chemicals that break dormancy. In ecosystems where fire is a regular occurrence, this adaptation ensures that seeds germinate after a fire, when competition from other plants is reduced and nutrients released from burned vegetation are available in the soil. Once dormancy is broken and environmental conditions are suitable, germination begins. The first step is imbibition, the uptake of water by the dry seed. As water enters the seed, it rehydrates the cells, activates enzymes, and swells the seed, often causing the seed coat to rupture. Metabolic activity resumes and stored nutrients such as starches, proteins, and lipids are broken down to provide energy and building blocks for the growing embryo. The embryo consists of a rudimentary root called the radicle, a shoot, and one or more seed leaves called cotyledons. In most seeds, the radicle is the first structure to emerge, growing downward into the soil to anchor the seedling and begin absorbing water and nutrients. The shoot then emerges, growing upward toward the light. In some species, the cotyledons remain below ground, providing stored nutrients to the seedling. In others, the cotyledons are pushed above ground and turn green, functioning as the first photosynthetic organs until true leaves develop. The transition from seed to seedling is a vulnerable time. The seedling is dependent on the nutrients stored in the seed until it develops functional leaves and roots. It is susceptible to desiccation, herbivory, disease, and competition from other plants. Many seeds germinate, but only a small fraction of seedlings survive to maturity. This high mortality is why plants produce large numbers of seeds, ensuring that at least a few will successfully establish. The longevity of seeds in a dormant state varies widely among species. Some seeds remain viable for only a few weeks or months and must germinate quickly or perish. Others can remain viable for years or even decades. There are documented cases of seeds germinating after more than a century of dormancy, having been stored in seed banks or buried in soil. The record for seed longevity is held by sacred lotus seeds, which have germinated after more than 1,000 years, though such extreme longevity is rare. The mechanisms that allow seeds to remain viable for long periods involve the maintenance of cellular integrity and the prevention of damage from oxidation, desiccation, and other stresses. Seeds are typically very dry, with water contents of only 5 to 10%, which slows metabolic processes to nearly zero and reduces the risk of damaging chemical reactions. Protective compounds such as antioxidants are present in the seed tissues to neutralize harmful molecules. The seed coat provides a physical barrier against pathogens and environmental extremes. For a listener seeking calm, the concept of a seed waiting in the soil, dormant and patient, offers a deeply peaceful image. The seed does not struggle or strive, it simply waits, conserving its resources, maintaining its viability until the conditions are right. When those conditions arrive, the seed responds not with haste, but with a measured unfolding of growth. Water is absorbed, enzymes are activated, the radicle pushes downward, the shoot emerges upward, Each step follows the one before in an orderly sequence governed by genetic programs and environmental cues. The patience of seeds is a reminder that timing matters, that waiting for the right moment can be more important than acting immediately. It is a lesson encoded in the biology of plants, refined over millions of years of evolution. The seed that germinates too soon may not survive. The seed that waits, that endures through cold and drought and darkness, may find itself in a world newly prepared to receive it, where light and water and nutrients are abundant and growth is possible. As this long exploration of plant and tree science draws to a close, the journey has moved through many quiet processes. The underground communication of roots and fungi, the slow drooping of branches at night, the patient capture of light in chloroplasts, the balanced exchange of gases through stomata, the ancient rings in bristlecone pines, the towering reach of redwoods, and the waiting stillness of seeds. Each of these processes unfolds on its own time scale, some spanning seconds, others spanning millennia, but all sharing a quality of steady, unhurried persistence. Plants and trees live their lives in slow motion, measured in the gradual expansion of cells, the silent movement of water and sugars, the incremental addition of growth rings. They are rooted in place, yet they are constantly responding to their environment, adjusting their growth, their metabolism, their defenses. They communicate through chemical signals, they cooperate through shared networks, they compete for light and water, and they endure through cycles of day and night, season and season, year and year. For anyone who has listened through this script, the hope is that these gentle, factual details about plant life have provided a calming backdrop, a slow stream of information that supports relaxation and invites sleep. The science of plants is rich and deep, filled with processes that are both simple in their principles and intricate in their execution. It is a science that rewards patience and careful observation, and it connects us to the living world in ways that can be both humbling and inspiring. If this quiet journey through the world of plants and trees has helped you feel calmer, or if it has accompanied you into rest, then it has served its purpose. If you found this content valuable, please consider supporting the channel by leaving a like or subscribing. Your support helps make more of these soothing, scientifically grounded explorations possible, allowing the channel to continue offering gentle, informative content for relaxation and sleep. Thank you for listening, and may your rest be deep and peaceful.